I'm Dr. Jim Dwyer, and with me today is Dr. Henry from Minneapolis Heart, I believe. And we're here to talk today about the ST-DETECT trial, which I sat in on this morning, thought it was very, very interesting. So if you could tell our audience a bit about that and what you found.
I'd be delighted to, Jim. So the idea that is very attractive is if we could recognize when a patient is having an ST elevation MI or even a non-ST elevation MI early. So if we had a way to have electrograms that we could identify in high-risk patient populations and then have a warning that would go either to the patient or the physician and that would have high accuracy, that would be a great thing. So the ST-DETECT trial was really based on that, and it was taking advantage of a patient population that was going to have an ICD placed anyway. Mm-hmm. And so the trial was sponsored by Medtronic, and so the patients in the trial were all met clinical criteria to have an ICD in place. So therefore, the majority of them had low ejection fractions. The average ejection fraction was 30%.
Were they all coronary disease patients?
They were. Okay. And so we took ICD patients who had known coronary disease, and our goal was to pick at least the goal of the trial was to pick high-risk characteristics. Patients with getting an ICD who had a previous myocardial infarction, who had 3-vessel or multi-vessel coronary disease, had positive stress tests, or who had previous PCI. So thinking that this is a higher-risk population for having subsequent myocardial infarction.
How was the ST segments detected? It was between the lead and the device itself?
Right. So there was actually two. The advantage of this is that there's two things. There was two vectors that were looked at, and then we looked at both sensitivity and specificity on both. And that is from the lead itself to the ICD, and then also from the SVC to RV lead. So there's two vectors that were actually potentially utilized. And
what did you guys find?
So the interesting thing is, this is a very well medicated and treated patient population. So more than 90% of the patients were on aspirin, many of them dual antiplatelet, around 90% on statins, beta blockers, and ACE and ARBs. And even though the left ventricular ejection fraction was 30%, the patients did remarkably well. We had 175 patients, and the trial stopped early because of low events. So even in this large trial, there was not one ST elevation MI. And there were 5 non-ST elevation MIs, and most of those were, 3 of them had no EKG changes at all. And there were only 9 unstable angina. So all of these low event rates, if you think about it, 0 ST elevation MI, a mortality of about 2%, a non-ST elevation MI of about 2%, extremely low. So really, unfortunately, there were not a lot of events to actually see if the technology works or doesn't work. We know that the algorithm was in there and was able to detect ST changes, but because the spontaneous clinical events were so low, we really weren't able to test the hypothesis sufficiently.
So what do you envision the future to be for this technology? Is it still available?
Yeah. So the second part of this trial is that there was a correlation, there will be correlation with stress myoviews. So we'll have that information that's going to be developed. And what I reported on today was really just the spontaneous coronary events. The second thing I think important point is I think we learned that this patient population with ischemic cardiomyopathies would be very challenging because the number of ST elevations are so low. Excellent medical therapy, and if you think, most of these patients have had bypasses, previous infarcts, so the risk territories for a new infarct are actually quite small. And when they did, they were non-ST elevation MIs, small enzyme bumps in small risk territories. So it still is a very attractive idea to be able to detect it. And I think the trick will be to do a trial design in a patient population that has a high enough event rate. And the good news, I think, for everyone is that we're doing extremely well with secondary prevention. I mean, so you can't feel bad about a trial in high-risk people that had no STEMIs. And I think, that is a testimony to really where, cardiology is today.
I think it's a brilliant technology, and I look forward to see where it takes us in the future.
Great.
Thank you very much.
Thanks for being here.